Good morning, good morning, good morning. Say somebody shoot me an okay on the audio visual stuff this morning. I'm going to try this without the headphones and see whenever I turn my head if you guys can hear me. So shoot me something on the live chat telling me yes or no, and we will adjust accordingly. Sound is good. All right. Video's okay too, I'm assuming, eh? Uh, So what you'll need to do is pull up the handout that I sent. I believe I sent it last night. It's on making a diagnosis. We're going to take a look at that today and think about uh, how how to go about making a diagnosis whenever we have a situation with a single animal, a single patient, or a group of patients, a group of animals that are undergoing that have had some issues and some challenges. So uh, before we get into that, and while we're still getting a few people coming on board here this morning, we're going to uh, take a look at some housekeeping stuff quick, and then we'll move into the information. Of course, this housekeeping stuff, you need to keep up with it because if you're not watching the lectures, then there's not enough people watching the lectures, the last lecture. So hopefully you can get caught up pretty quick. And it's important to see the material and hear me talk about it. And also this housekeeping stuff saves a lot of questions and so on and so forth. So okay, if you haven't got homework 6 in yet, most of you have. Be sure you jump on that and get that to me. The presses on cheatgrass and fire rehabilitation and grazing and so on and so forth. So get that to me if you haven't done so. I'm going to try and hopefully I'll get them graded over the weekend. I hope I'll have time to get through them. And be sure you check your email I sent in this morning to you. It has a link. Well, it has a couple of things on it. One, it talks about exam 2 a little bit. I'm going to talk about that a little bit right now, and then I'll come back to the Yes Awaken link and that assignment. Exam 2, I'm going to hand that out on Monday. It'll be an open book type test. It'll be multiple guess. And along with the exam, I will be sending an answer sheet. And that answer sheet will be in Word format. And it's a pretty new Word format. So hopefully everybody will be able to pull it up and look at it. Both the exam, I'll send the exam and I will send the answer sheet, both probably in Word format. But the answer sheet for sure will be in Word format, and you need to fill in the answer sheet very cleanly and very concisely. So if I send it out in PDF, not everybody will be able to input anything into it. So it's coming out in Word. You can put your answer down where it's supposed to be and then save it and send it back to me. I don't need the exam back, but I will need the answer sheets back. And so I'll talk a little bit more about it on Monday. I'll either send the exam out over the weekend or early Monday morning so that you'll have it in time for class and just in case you have any questions about it. But I'll go over the answer sheet once again. That's what you're going to need to do is fill it out in Word and then get it back to me. So that'll be exam 2. That'll be next week. On Wednesday of next week, we will not hold a lecture period, a live lecture period during this regular lecture scheduled hour. And so you can just use that if you haven't already used it. Just call it an exam day, and so we won't meet on next Wednesday, which I believe is the 22nd of April. So don't expect me at 8 o'clock to 8:50 next Wednesday. Just use the time if you to work on other stuff, work on your exam. Okay. Um, Also, on Monday next week, we're going to, and that also pertains to the email I sent out this morning, we're going to discuss this video performance of the, I don't know what to call them, super group rock band from the late 1960s. They're still hanging around and doing some pretty good stuff. But they had this particular performance from the early 2000s. I want you to have a look at it. And we're going to think about things in terms of professionalism. We're going to use that video as an entree into discussing professional behavior and professional content, those kinds of things. So be sure you look the video over. If you're having trouble with the link, drop me an email, let me know. It should be okay. It's a YouTube link. Put on your, take some time to concentrate on it. Go, be by yourself. Don't be watching a movie while you're watching it. Put your best buds and your headphones in and really kind of try to absorb it, because I want you to, and while you're doing it, I want you to think about what went into that production in terms of professional behavior, so that you can identify those things in your own practice and in your own day-to-day professionalism. So we're going to talk about that on Monday. So I want you to watch it over the weekend. You may want to watch it a couple of times. We're going to talk about it a little bit on Monday. And then we're going to talk about it again on Friday. Okay? Because what I'm going to do is, after we talk about it on Monday, I'm going to send you home. I'm going to make you watch it again, thinking about what we talked about in class and what you write down in class. And then we'll come back and we'll talk about it again on Friday. Of course, this will be through the live chat sequence. And so your professionalism discussion, what I'm going to want is a write-up on this these discussions and what you think is professionalism, and basically your task will be, sort of as a professional the rest of your life, is to search for that moment when you are absolutely the best you can be at that particular task or role that you're involved in. It doesn't make any difference what that might be. But that's what you're trying to do with professionalism, is really search for that moment. That very moment. It's a constant search, but it's a search for that very moment in some task or in some role. That moment when you are absolutely as good as you can be at it. Okay? And so I'll give you some outlines on how many words and what the format kind of stuff is a little bit later on. I don't want you to get too bogged down with that right now, but that's the purpose of this. And so I want you to take a look at that video. Hopefully you'll get through it maybe 3 or 4 times before you sit down to write. And while you're thinking about that professional approach to your job life, although a lot of us don't have job lives right now at the moment. But that's the point of the assignment, and like I say, it's going to be due on the 29th of May is what I have on my calendar there. So we'll talk more about that on Monday. And hopefully you will enjoy enjoy the process of doing that. And so it's a little unconventional, but and maybe it's strange and unusual. But like my wife says, I am strange and unusual. So I think you'll find it a very beneficial exercise. So how are your interviews progressing? You can't all talk to me on live chat, but I'm getting some feedback from some of your interviewees, getting some good feedback so far. So if you haven't been involved, haven't done your interviews yet, some of the feedback I'm getting back is pretty good feedback. So you've got the bar is high. So be sure you meet that bar when you're dealing with your interviewees. So no one's having any trouble setting up times with them. I would say the sooner the better, because if and when these travel and work restrictions, COVID-19 restrictions are lifted. If they are lifted at the end of the month, most people are going to, they're sort of living a nightmare now. Wait till all backup stuff starts happening that has been pushed back. So people's time is going to be very, very thin, and so don't wait. Get to it and get it done would be my suggestion. The sooner the better. Don't sacrifice quality, but the sooner you get it done, the better off you'll be. Just in contingency, and of course that's professionalism. Always have a contingency plan. Always plan ahead and think what might happen in the future. Okay. So I think that's all the housekeeping stuff I have for you this morning. Yeah. Okay. Let's take a look at the handout that I sent you. If you haven't pulled it up already, pull it up. Some of the photos may be a little disturbing for some of you. So if they are, don't look at them for very long. But I do want to kind of go through this and give you an idea about how to go about, some of the things, not all of the things, but some of the things that go into making a diagnosis. So that's the title page there. We're going to look at some poisonous plant stuff, and we're going to look at forage kochia, which is also an issue in Nevada. Although it's not poisonous, it gives us some other problems. So we're going to look at that over the next bit here. We may look at it a little bit on Monday too, depending on how far we get. So your second slide there, second page, number 2, I think I sent it to you as a PDF file. The PowerPoint's a little bit too big to go over the email cyberspace, but what you're looking at in slide number 2 here is a problem that's been around in the Intermountain West since the time that we brought sheep in, back in the late 1800s and early 1900s. And there was this condition. Sometimes ewes would give birth to lambs that you see down here in the lower right-hand corner. It's a condition that was called monkey face lambs, still called that. And we didn't know. Nobody knew what was going on, and it didn't happen every year, and it didn't happen every year when it did occur at the same rate. But it did happen enough across the whole entire Intermountain West that it became an issue that the Agricultural Research Service and the Poisonous Plant Lab over in Utah became involved with it to try and find an answer to this condition called monkey face lambs. And so if you look at you see there's an older picture there. You can tell that the other slide of that ewe standing there has been there for a while. So this had been a problem for a number of decades, and finally in the 1950s ARS started working on it, or ARS, the Poisonous Plant Lab, in conjunction with ARS, they began to work on it over in Utah, the Utah station. So if you look at the next slide, what you see is a picture of this very, very large-leafed, succulent plant. And so you have to have a place to start. And so what the scientists did is they started talking to sheep herders. Most of them at the time were Basque sheep herders, a few Irish sheep herders around, and they would ask them, they said, what do you think is causing this problem? And they would tell them, almost all of them would say, we think it's this plant that's causing it. And this plant, the common name of it is skunk cabbage, is the name of this plant. And it grows in sort of higher elevation mesic areas where you tend to see sheep in the springtime. And so this is a great sort of habitat. You see aspen in the background there for milk fat lambs on Forest Service country. And so this is a relatively common plant that occurs. It's not as common as some other poisonous plants, but it does, it's not rare, it's just infrequent. But the sheep herders would, they were telling them, we think it's this plant. And how they knew that, nobody really knows. It's just sort of an instinct. When you live with animals, you get to thinking about things and you come up with ideas. And anyway, that's what they thought. And so the plant lab, in the next slide, they decided, okay, let's try it and see if they know what they're talking about. It's a place to start. And so they started harvesting skunk cabbage by the truckload. Yeah, I was going to say buttload, but by the truckload. And they started gavaging pregnant ewes, and even some non-pregnant ewes before they actually became pregnant. Anyway, they started gavaging these animals, and with a concoction of just almost pure, skunk cabbage and water. And they did this literally for years, and they could not get an effect. And so they go back to the field, they ask the sheep herders, they say, are you sure? And they say, yeah, we're pretty sure it's that plant. And so they began this process of gavaging these animals at different days, on different days during gestation. Okay? And when they started doing that, because otherwise they were just dumping it in, and they couldn't get anything. It didn't bother the ewes. They're not poisonous to the ewes, and so they started doing it on a sort of an organized basis, where we would do it on this day of gestation, then we would do it on another day of gestation, and then another day of gestation. And finally, after a number of years, what they finally found was an effect, and they started getting some lesions. But the problem is that I'm looking at slide number 5 now, page number 5. If you go to page number 6, what you see there is a spectrum of lesions from the same gavage concoction, but it's based on a single day of gestation, and it's based on whether they were gavaged at 1 hour or at 23 hours on that one particular day of gestation. So you see, they just kept gavaging all the way through gestation at first, and then it turns out it's only like 24 hours on a single day, on a single certain day, when the cephalic portion of the fetus is in development, or is in a certain stage of development. And so they kept missing it for years and years. And finally they hit on the day, and then they found out that it made a difference what the severity of the lesion was, depending on whether they were gavaged in the morning or gavaged in the evening, so to speak. So, okay, you with me? And so they finally figured it out. And so here's a whole spectrum of here's some from cattle, or not cattle, sheep. A whole spectrum here of sheep being gavaged with this stuff, where you have almost a lamb that is not affected very severely at all the way to sort of this hydro or this cycloptic, severe cycloptic lesion. And if you see there at the bottom on the right, that is the most severe lesion that they could induce. The head and brain is just not developed at all. And so they were able to induce this entire spectrum of lesions based only on the time of a single day in gestation when these animals were gavaged with skunk cabbage. So sometimes making a diagnosis can be very, very complicated and complex, and sometimes you just have to get lucky in order to do it. But this gives you an idea of how difficult trying to make a diagnosis can be sometimes. It has been at times. So, and it turns out that this particular effect is also something, if you look in the next slide there, slide number 7, is very similar to some effects that some children were being subjected to in other countries, in developing countries, where their diets were such that only a single, if the mother consumed a certain product or certain plant type on a certain day during her gestation period, then you could see some of these same cycloptic effects in human fetuses. So the monkey-faced lamb work bled over into some human work, and they finally discovered some things that were causing this in some developing countries across the world. So, so your next slide there, number 8. I mean, this sort of explains a lot when it comes to veterinary practice, whether it's cows or dogs. You know, they're eating mostly cactuses. They're a toxic principal in cactus that could account for their poor condition. You know, and so then, you know, the concept is Christ, if they're already eating if that's what they're on and they're eating cactus, something else is wrong. It's not the cactus that's the problem. It's a management issue at that moment in time. So yeah. All right, next slide here. I've got slide number 9. You've probably seen this one earlier in the semester. Diet preferences can be pretty complex. They're behavioral, they're chemical, they're habitual, and it depends a lot on the palatability and the nutritional quality of the plants that animals have available to them, whether they will eat them or not. The next slide is just a bowl of some stuff. It's some kind of salsa. And if I look at that, that has got to be the crappiest salsa I've ever seen in my life. There's no way that can be any good. It just can't be. And yet, that's my diet preference, just my visual acuity of that. I look at it and I go, nah, give me some other salsa. I don't want to eat that stuff. So it just goes back to that diet preference thing when it comes to animals and what they may eat, what they might not eat, and all the reasons behind why they may and may not eat something. Sometimes animals are sort of captured and food has to be brought to them for the most part. And so we have to be careful about the quality of the feed that we bring to animals, particularly when they're large, expensive companion animals, we have to be cognizant of the feed that we're feeding them. Lots of people have in ranchettes, they don't have enough land to feed horses on grass naturally, so they have to introduce feed like hay or oats. And so we have to be careful about what we present to them in these kinds of situations. The next slide, number 12. This is an example of a sheep die-off from the early 1940s. We're not sure whether this is the late 1930s or early 1940s. It is in color, but this is an example of a sheep die-off from Halogeton, which is a range plant. It's an introduced annual chenopode range plant. Grows all over the place in Nevada and throughout the Intermountain West, right on the edge of salt desert shrub type plant communities, which is typically sort of a winter range and very early spring range for sheep and for cattle. And so we would have situations like this where somebody might have an entire, particularly on a drive, whenever you're moving sheep, you might lose 800 or 1,000. You might lose a whole band of sheep on Halogeton. And Halogeton has some poisonous aspects to it, oxalates, which will kill animals. But what we learned is that it was more of a management problem than anything, because when we would drive sheep down some of these drive lanes that we have in the Intermountain West, and we had them moving all day, and then we'd get them someplace to where they could water in the evening and then bed down. They would pick up Halogeton around these water sources, and sometimes even on the way, and then guzzle a bunch of water and then sit down, and it would exacerbate the effect of the oxalates. And you wake up the next morning and you have 500, 600, 700 head of sheep that had tipped on you. And that is a gruesome sight. That's not something I would wish on anybody to have to see. But we found out it's more of a management problem. It has to do with how much of the Halogeton an animal consumes, and how quickly they consume, and whether you can buffer it with something else. And maybe you don't want to bed them down in a certain area around the water. Maybe you want to move them off a distance and bed them down. So it becomes a management issue there. But it turns out in this case it was a Halogeton. You can see the Halogeton there in the picture, that little straw-colored plant that's sticking up, has a stem sticking up all over the place there. That's the Halogeton that they've gotten into. The next slide is, somebody just sitting there trying to figure out what happened to critters. And so you just have to kind of stop sometimes and think about the environment that the animal's living in, and what you see, what is the evidence, and you try to put two and two together. It becomes sort of a puzzle of trying to piece together what may have happened. The next slide there, number 14, let's just take a look at this real quick and let's see what we have. We've got at least 3 dead yearlings laying here in an area of green. We've got aspen in the background. The aspen look like they are just putting out their leaves, so it's early in the spring, or very early summer at whatever elevation this is. It's at least at aspen elevation anyway. What could this be? And usually, the first thing that students want to talk about is snakes. Is there any snakes there? Well, yeah, there may be some snakes there that early. They're not going to be very active in the morning. They're only going to be active for an hour or two a day because it's still too cold. But what is the likelihood of a snake or a group of snakes, biting at least 3 yearlings? So it's probably not snakes. It's not predation. They haven't been attacked by a predator. So what would you suspect? Well, the first thing you would think about then, ruling all this other thing out, the first thing that makes any sense is they've gotten into something poisonous. Maybe it's a poisonous plant issue. And so if you look at the next slide, you'll see a picture of sort of what's growing around there. And sometimes you have to actually go out as a practitioner, as a rancher, or as a manager, or as a vet. You have to go out and you have to kind of physically look around and see what's going on there. And so if you went out to this area, this is what you would have seen on that particular day. And what do you see here? Well, I see some geraniums, a little cut leaf forb there. I see some grass growing around the area, and I also see one other very conspicuous thing there. You'll see it over in the upper right-hand corner by the cow pat, sort of a grass-like looking plant there, and that is death camas. Okay, death camas. So don't ever eat anything that has death in the name of it. It's not going to be good for you. And if you look over in the lower right-hand column, you'll see some death camas there that has been bitten off. And this stuff is so toxic. It just doesn't take more than a few bites, 4 or 5 bites of a decent-sized plant, and you're going to have issues. You're going to have mortality issues with cattle. But here we got something growing. It's green. It's sticking up. It's a grass-like plant, and there's not a lot of green out there. In fact, this is usually one of the earliest plants that greens up, and so cattle they'll just clean it up. And if they don't have anything else to buffer it with in their diet to any significant degree, you're going to have issues, and you may have issues anyway. So we have to kind of clock this up to death camas. We have other poisonous plants, and I hope to visit with you some more about that before the semester's over. This is larkspur. We have a number of different kinds of larkspurs. We've got some perennial larkspurs and some annual larkspurs that grow in the Intermountain West and around Nevada. I think the annual ones actually are more toxic than the perennial ones. We have larkspur that animals can get into. We also have senecio that animals can get into and have some trouble with. And then we've got range animals and we've got confined animals. I mentioned confined companion animals a while ago. The next slide here, slide number 19 is what I'm looking at. We're looking at some oats and we're looking at some alfalfa cubes and some hay bats there. We have to be cognizant that so that we don't bring toxic things into animals that are being raised in a confined area. And we can do that when we bring any kind of these feeds in. We have to be careful about our source and about the quality of the feed that we're bringing in and presenting to animals. Here's an example of a feed or of a poisonous plant that can get into oats. If you go buy oats for your horse, if it's not quality, if you don't know who you bought it from, some people just go and buy it at the feed store. That may be all well and good. Sometimes it's not. The feed store may have gotten it from a local vendor. You just don't know. But this is a plant on the right-hand side here is Jimson weed. You've probably heard of that plant, Jimson weed. And some of you may take a little bit of it. It's a what do you call one of these over-the-counter supplements that you can get and that you can take. But Jimson weed. If you've got Jimson weed growing somewhere around your oat field, and you get some of these Jimson weed seeds, if it's growing in the oat field, and the combine comes by and harvests the oats, it also harvests this plant, harvests some of these seeds, and you get just a handful of Jimson weed seeds in a mouthful of oats for a horse, and you're going to have trouble. You may have a dead horse tomorrow. So the quality of the feed is very, very important, and you have to be cognizant of that. If you're going to go buy feed for an expensive animal or an animal that you care dearly about, Make sure that the quality of the feed is there and that you know the source of that feed. Okay. The next slide is a slide of some senecio growing right next to a barley field. This happens all the time. These weedy species get into these fields, and when they're cut for hay or they're combined for seed, uh, these plants can be transferred into that hay, can be baled into the hay, or the seeds can get into the product as well. Well, how can you figure it out whether you've got the quality or not? Uh, you figure it out ahead of time, but if it's one of these things where you're buying from a local or a regional vendor, sometimes you have to do some testing on the forage. And so here's an example of a homemade hay tester, where you just, for a square bale here, or you can do it with a round bale, you just take the truck and ram it with this pipe and pull some cores out and send them to the lab, have them tested. We have the ability to do that in Nevada, and so we have a testing lab that can test some of the toxic principles or toxic components of some of our feed. If we can't do it here, they can do it over at UC Davis. And so something to keep in mind if you're getting into veterinary practice or if you're getting into managing livestock at some point in time. The next slide, sometimes what you have to do is you have to revert to, chemical analysis, send it to Davis, send it to the state office here in Reno, or actually they're over at Sparks on the other side of Gregg Street over there. And sometimes they have to do some really, really fine-tuned chemical analysis in order for you to get a diagnosis. So sometimes the science really has to be there for you to get a good diagnosis or give you information to make a diagnosis with, I should say. So that's what the next slide there is just an example of some laboratory tests and whatnot that may be important. The next slide I've got here is slide number 25, which is this goat. And so I usually ask the question, what do you see here? What do you see that's unusual about this goat? And of course, everybody wants to say, well, it looks like they nicked itself shaving or something, right? But that's not what I'm talking about. What I'm talking about is, and people call it a devil goat too, but what you're talking about is the animal behavior. You can tell from this picture, if you know anything about goats at all, if you have any background in goats, what you can tell is that this goat has what is called a stilted gait. Stilted gait. Its legs are stiff. Look at its tail stuck up in the air. You know right away that something is wrong when you see something like that. So just an observation of animals can tell you a lot about what's going on with them, whether there's something really wrong with them or not, or and so familiarize yourself with animal behavior and how they carry themselves, how they stand, how they hold their ears. When that old mare's got her ears back, don't go in the pen. But when she's sort of chewing with her lips, she's happy. So notice those types of behaviors with the animals that you're working with. It'll help you figure out a diagnosis. The next slide here, which one is it? Number 26. This is just a slide of a postmortem. Sometimes you have to go to a postmortem, particularly if it's a chronic. If you can't, a chronic issue. If it's an acute issue, sometimes you can figure that out. If it's a poisonous plant, you can usually figure that out without having to get to a postmortem. But there are these chronic issues that develop with animals, and the only way that you can really, in lots of cases, not always, but in lots of cases, the only way that you can really determine what went wrong is through a postmortem. And so this is just an example of that. The next slide, uh, you're looking at some lungs here, and right away you can tell that something's been eating holes in the animal's lungs, and so that's not good. But you can find out what critter it is. Maybe it's a lungworm in this instance right here. So you're looking at you can find the lungworms actually in the lungs, and you can make that diagnosis. Um, Sometimes you have to go to making slides of tissues, particularly the liver. Remember, the liver is one of these organs that detoxifies most of the toxic materials that animals take in. Goats have a big liver, and so sometimes you have to do slides uh, and look at them under a microscope in order to determine whether there's, for instance, there's a blast here where where the arrow is pointing, where you have a parasite encapsulated in the tissue. So sometimes you have to get to post-mortem situations, and you have to really sort of fine-tune the post-mortem situations. So here's your map to a diagnosis. Your map to a diagnosis. Are there clinical signs in the animal that you're concerned with? If they're dead, that's pretty clinical. Are there clinical signs? Are they, do they have snotty noses? Are they limping? Do they have a stilted gait? Are they pressing their head against the fence? Are they depressed? Do they have anorexia? There's any number of clinical signs that you can look at with animals to determine, that will help you determine that diagnosis. So that's one of the first things you want to look for is clinical signs. Study results. Go back to our skunk cabbage issue. Are there, look at what are they exposed to in their diet? You need to know something about plant identification, at least a very, very cursory bit of it, in order sometimes to make a diagnosis. And you may have to resort to chemical analysis of the forage, or quality or chemical analysis of the forage that you're feeding them, whether it's purchased hay, or purchased concentrates, or whatever the case may be. Blood and biopsy studies. You may have to do that to find a diagnosis. And you may have to get to where you're actually having to perform a necropsy and doing some histological studies to determine a diagnosis. So you take in all the information that you can take in, make a diagnosis, and once you get a diagnosis, then you can prescribe treatments for that particular issue. So you, as the manager, whether you're a veterinarian or you are a herd manager of some kind in a ranch type situation, you are the decision maker. And so you collect information to make decisions on. Okay. And I was listening to our, just an example of what I'm talking about. I was listening to our governor yesterday afternoon, and he is right, wrong, or indifferent. I'm not taking a side on this, but he was intent on saying that his medical team, he's listening to them making his decision. Well, that's all well and good, and that's what he should do. But he's allowing then he went on further to say that they are going to tell me when it's time to turn the spigot back on. Well, no. He's the decision maker. He listens to the information, and then he makes the decision. So if you're a veterinarian and you have to send stuff off to a lab to get some information to make a diagnosis, and the lab technician tells you something, you may agree with it, but you ultimately are the decision maker. You can't be letting the lab technician make a decision for you or make your diagnosis for you. You are the decision maker in that instance. And so this is sort of your map to a diagnosis here. It doesn't always work like this, but this is a pretty generalized approach to try and figure out what is going on out there. Okay, So let's look at the next slide. We got a few minutes left here. It's a resource list for you that you can use in the future. A number of things there that are hopefully will be helpful for you. So let's look at the next slide past the resource list, slide number 31. Let's talk about a few little incidences. And in fact, let's go to slide number 38 or page number 38. Let's look at that. And we'll see what we have time for on Monday. But I want to introduce you to this one. This is not. This is an exit. What we've looked at before are toxic plants. What I want to look at now is a plant that's not toxic, but gives us all kinds of trouble in certain years with domestic cattle. Okay, so if you're looking at page number 38, excuse me, what you're looking at is a forage kochia seeding. A forage kochia is a sort of what's called a subshrub or a half shrub. It's from Central Asia. It's not native to Nevada and the Great Basin, but it's being used in fire rehabilitation all over the Intermountain West. And it has some great qualities about it. It's great feed. It's high protein feed. It greens up in the fall when most things are beginning to senesce. It's readily eaten by cattle and sheep as well. And it's a great fire rehab species because it takes hold very quickly. And it sort of takes the place of sagebrush as an intermediate kind of a species. And so it's being used literally on hundreds of thousands of acres across the Intermountain West. And it's a great, I mean, it's a great plant. It does a lot of good things. But we began to get some calls a number of years ago. It's been almost 10 years ago now. We started getting our first calls, and we started getting people ranchers started saying, wait a minute, something's not right here. We're starting to get some animals that are dying and they've been eating this stuff. Is it poisonous? And so we go to looking, and I'm looking around and I'm making phone calls. I'm going, no, nobody's ever heard of this stuff being poisonous at all. I mean, it's forage in Central Asia. It's a common forage over there where it came from. They use it all the time. Been using it for thousands of years. And nobody's ever reported it being poisonous before. And so instead of being poisonous, what they were dying of, and we had a handful of them posted in the field, did some necropsy work on them, and these animals were all dying of frothy bloat. Frothy bloat. Now, frothy bloat is different from bloat. Frothy bloat makes a lot of foam. Bloat, you get a lot of bubbles in the rumen, but with frothy bloat, you get a lot of foam. And it's the same principle. The animals die of bloat, but it's a different type of bloat. And frothy bloat is based on a couple of things. One, high protein and saponin content, and the size of the forage particle. The smaller the forage particle, the more likely you're going to get frothy bloat. And so we started looking at it, and here's, if you look in slide number 39, this is a picture of somebody pulling a rumen plug in an animal that has frothy bloat. Now, can you imagine the distress that this animal is under to have a blowout like this? It's just incredible, the internal pressures that can form in a rumen. The next slide, slide number 40, is a slide of a rumen that is being subjected to frothy bloat, a color slide. And so frothy bloat, what I equate it to is like a root beer float, the head on a root beer float. Some of you will never drink a root beer float now again. But the head on a root beer float is much like frothy bloat. And the residence time of that froth is very, very long. In fact, what we found out in the lab is that for alfalfa, when an animal is bloated on alfalfa, it makes big bubbles, and the residence time of the bubbles are about 6 hours. But for frothy bloat, the residence time of the same uh, bubbles, the same bloat entity, can be 12 to 24 hours. And so it just does not go away. Uh, and so this has been a problem in falls, in the types of years where we have wet falls and the forage kochia really greens up and it's really going, and there's not a lot of cheatgrass or other perennial grasses around to buffer it. That's when we get a situation where we're getting these frothy bloat mortality incidents, and they are big. I mean, sometimes we're getting 20, 25 head of cattle that are tipping. But you can manage around it. You can manage around it by kicking out a little bit of hay, some dry hay, or maybe some supplement, or some bloat salt. Or you can put some bloat stuff in the water if you're hauling water. So you can manage around it. But it's just an example of a plant that caused some problems that we had no idea that it was going to cause problems. And we've got millions of acres, well, millions, we've got hundreds of thousands of acres of it out there now. That it's a great feed, but we have to just be cognizant that some bad things can happen. So, okay, time's up for today. I hope you have a great weekend. Be sure you get your homework in to me, and watch your Yes Awaken video. Look at it a couple of times and be ready to kind of talk about it a little bit. I'm going to ask you some questions on Monday, and we'll get the discussion started. So if you got any questions, hit me with the live chat. Well, they do have some other forages, but typically they don't have the quantity of forage kochia that we have. We seed it. We seed it a lot. And so the density of that plant is much less in Central Asia than it is where we actually go out and seed it with a drill rig. So why doesn't it happen today? That's sort of it. They don't have the densities of this plant in Central Asia in its native habitat that we have here. We actually go out with a drill, a range drill, and we drill this stuff. And so the densities are quite high, and that's one of the main reasons why they don't have any problems with it. And then it's sort of a seasonal range for them. They move animals through pretty quick, and they're on it and then they're off of it. So it's management, and of course availability. We have a lot more of it available here in lots of areas than they do in Central Asia. Any other questions? Going once, twice. Sold American. Okay. Have a great weekend, and we will see you and talk to you again on Monday. Bye-bye.